A bad concussion can raise your risk of Alzheimer's decades later. So can the genes you inherit. Two roads, one destination. No one could explain how a physical hit and inherited DNA arrive at the same disease. I wanted to know if they run through the same wiring inside one type of brain cell. That cell is the microglia, the brain's resident immune cell, and it's where Alzheimer's genetic risk concentrates. Based on a deep literature dive and working only from public data, I defined two opposing gene programs in advance before looking at the results. An accelerator, the pro-inflammatory program, and a brake, the pro-resolving program built on TSG6 and hyaluronan. Then I tested whether injury and inherited risk both hit the same one. I checked it 4 ways in human and mouse: single nucleus RNA from microglia in Alzheimer's brains and injured mouse brains, bulk RNA from sorted astrocytes, spatial maps that keep each cell in place around plaques and around the injury lesion, and brain proteomics, 9 analyses in all. The accelerator converged in both, every way I looked. In brain injury and in Alzheimer's, the microglial accelerator is floored while the brake fails to engage, and it concentrates exactly where the damage is, beside the plaques in Alzheimer's and at the lesion in traumatic brain injury. Then the genetics gave direction. 31% of inherited Alzheimer's risk sits in 1.5% of the genome, a 21-fold enrichment, and it loads onto the accelerator while avoiding the brake. Here's the sharp part. When I tested whether those variants control the inflammatory effector genes, they didn't. Inherited risk enters upstream at ApoE and TREM2. Downstream genes like SPP1 are installed by the injury or the disease itself. Threshold and trigger are separable. The two-hit model made molecular. Both arms are read out at one receptor, CD44, and it isn't only expressed on microglia, it's also on astrocytes that ring the plaques. CD44 rises across 4 different measurements and in both species, while the brake ligand TSG6 falls. One receptor decides the outcome. This is where the rubber hits the road. CD44 carries both signals, so blocking the receptor fails. You'd silence the brake along with the accelerator. Instead, rebalance the hub 2 ways: block the inflammatory ligand to remove the push, or provide TSG6 in trans to restore the missing brake. Each is a ranked, testable prediction, a specific experiment. For decades, we've treated neurodegeneration like a runaway train. Maybe the answer isn't stopping the engine, it's giving the brain back its brakes.